Okay, everyone. Well, we might get started bang on time. So welcome to our third OHBM Australia webinar. We've got a great program lined up today. I just wanted to give you a brief update of the chapter's activities, just so you're aware of what's been happening, and then we can jump right into it. So this year we were planning to hold an in-person meeting, and it was going to be a joint meeting with the Australasian Cognitive Neuroscience Society and the Experimental Society for Experimental Psychology. And that was going to be in Brisbane, but obviously due to COVID, we needed to put that on hold. And just to kind of update you that the intention is hopefully to still aim towards an in-person meeting next year, again with the societies all together. Obviously, it depends on restrictions. Things are going well at the moment, but there is still a lot of uncertainty. So at this stage, we are looking holding it towards the end of next year, but we will make announcements as the picture becomes clearer throughout the next year. That said, in the meantime, the chapter still intends to hold virtual webinars such as this one, and we'll do that on a kind of semi-regular basis every couple of months or so. We asked everyone to complete a questionnaire as to the topics that they're interested in, and so we're going to work through that list in rank order. And the sort of top-rated topic that came up from that questionnaire was how to process MRI data. So everyone's interested in pre-processing. And so we structured today to really focus on, what are two of the most widely used modalities, particularly for mapping brain connectivity, and that's diffusion MRI and functional MRI. Now, in each case, the analyses rely on pretty complicated multi-step processing pipelines, and at each step you need to make a number of different decisions. It's not always obvious which decision is the best one to make, but the decisions you make can affect your final results, and every week we're seeing new analyses showing the impact that these choices have on analysis findings. And so it's really difficult for someone who's coming into the field to know what to do, how to, to know how to make sense of it. It's really become a sort of specialist area in its own right. So the intention today is really to discuss progress towards establishing best practices for the field. And, that's really providing perspectives of innovative researchers that are really pushing the boundary in the area, as well as EMCRs that have been very active in these fields. And so the first half of today's session is going to focus on diffusion MRI and is going to feature a keynote from Maxine Decotter. I hope I've pronounced that correctly. I've probably butchered it. Followed by a presentation by Maria Dibiase from the University of Melbourne, and then an open discussion moderated by Andrew Zaleski. And then the second half of the session will focus on functional MRI, and it features a keynote from Cathy Chang of Vanderbilt University, followed by a presentation from Sasuke Balman of University of Queensland, and then an open discussion moderated by Adil Razi. So we hope that this webinar will be both educational and informative, and will push you to think carefully about how you process the data. With that said, I'll hand over to Andrew.
Thanks, Alex. I'm Andrew Zaleski. I'm moderating the session on diffusion MRI. We have two wonderful speakers lined up for this session. We have Max Descatou, followed by Maria Dibiase, and then we'll have a discussion at the end of the session. Max is internationally recognised for his work in diffusion MRI, particularly in tractography. He's developed novel tractography algorithms, as well as developed phantoms to evaluate these tractography algorithms, and also drawn attention to some of the key limitations and challenges of performing tractography using diffusion MRI data. So Max leads and established the Sherbrooke connectivity imaging, validating and using human brain mapping and connectomics applications. Max holds the University of Sherbrooke Institutional Research Chair in Neuroinformatics, and his work has been cited more than 8,000 times, and he's published more than 105 journal publications. Really looking forward to your presentation, Max. So over to you.
Great. Well, thanks, Andrew, for this nice introduction. Really happy to be virtually with you guys in Australia. Wish I could travel and be with you in person, but this will have to wait for a little while. So today I really want to talk about, the road towards best practices or consensus. Probably have to talk on what I call diffusion pre-processing. So trying to get the best corrected diffusion data to which then we can start doing DTI, ODF, Tractography to build the connectome. So half pre-processing and half, let's say, post-processing towards Tractography. It's really hard to cover in 25 minutes, so it's going to be pretty packed, and we can leave some stuff for the discussion. So first thing I want to say is, towards a consensus was really a big part of my job as a president of the diffusion study group back in 2018. I took on myself to do this survey in the whole diffusion community, mostly on the ISMRM side. And overall, from 200 respondents, we got general trends that people thought consensus could start being reached. And one of the first thing we established is that the word consensus was probably badly chosen and best practices was better. And then 4 projects started from there. The first on DTI, that's led by Carlo Pierpaoli and 2 other colleagues. And then beyond DTI, a third one, which is clinical tractography. And the last one is diffusion pre-processing. And so you should see coming out in the next year, I believe in 2021, probably 3 or 4 papers out of the diffusion study group with 20 to 100 co-authors, trying to establish the best practices for clinicians, for the industry, for new students coming into the field. And so this is kind of good. Things are moving forward. And I want to thank Jelle Verraert. If you haven't done so yet, please fill out this survey. It's an ongoing survey about diffusion processing. So if you're involved at all with diffusion, no matter the organ, it's not just the brain, go and fill out the survey, give us your opinion, and this will serve in the process of trying to build this paper. So some of the slides I'm going to present today are from Jelle, so I want to thank him. And you might have seen them around at ISMRM or other conferences such as MICAI. And before I get into this, I want to say a disclaimer, there's tons of good software out there. A lot of public, very good MATLAB, Python, C++ software. So it's not going to be a talk about software. I'm going to point to a few software here and there when they have good solutions or when they were pioneers. But there are as many software out there as algorithms. So and I think there's a very nice push towards open science and reproducible science through these softwares. And all through the talk, so the new students in the room, keep track of my stars. Whenever you see a star, that means it's a really well recommended and probably established best practice for diffusion experts. So if you see a star, it's probably something, it means it should be part of your analysis pipeline. And if you see a warning sign, it's probably still open questions out there or considerations to be made. And so if your students also are developing new methods, this is probably areas where there's lots of work to be done and publications to be made. And before you start anything, and that's something I always tell my students, we want to crush numbers and start doing statistics and fine and beautiful results. But the first thing you need to do, and this is a big investment, but you need to do it at the start of your project because you will hurt from this if you don't. Look at your data. We never look enough at our data. And lots of software out there are available now. One of the things you should always look at in diffusion is your gradient table, the famous B values and B vectors. And here's just an example on a project I participated in Canada, on the Canadian Consortium on Aging. So we were planning to build a harmonized protocol across 30 sites in Canada, 3 Tesla scanners. We scanned the same individual 108 times across these sites before we launched the consortium on Alzheimer's patient. And it was a simple single shell, 1B value, 30 direction protocol. And after 2 years, I received the first data sets. And here's what I found. 3 different sampling schemes going on, each with 30 directions at B1000, but bugged sampling schemes, uniform, very good looking sampling schemes, or geometrical radial sampling schemes. So even though this was supposed to be a simple harmonized protocol across Canada, we managed to screw it up, and resulting consequence, if I look at 100 data sets from the same brain, the same guy, Simon Duchesne, the PI on this project, well, as you can see, I don't have time to present all of this, but there's significant differences between Siemens, GE, Philips, and for DTI measures, as well as Hardy measures, and pretty much everything. And so large variability, all of it explained for the most part because of these differences in sampling scheme. This hurts even more when you don't have lots of diffusion directions acquired. So keep that in mind. Have a look at your gradient scheme. Recently, I QC'd the whole Human Connectome Project database, and we found 81 subjects with wrong sampling schemes. Instead of 90 directions for B values, 1,000, 2,000, 3,000, we found sampling scheme with 60, 60, 60 that were not well distributed. So even the very good consortiums out there make mistakes. And if you just grab all this data and start processing, this may lead to high variability and to statistically significant results that are not driven by your research question. The other thing you should always look at, and I love these little movies, is have a look at an animated GIF of your diffusion weighted imaging. You right away see motion, you see the head tilting, you see the eddy currents, you see a little bit of artifacts. In this case, nothing we cannot deal with, but if you have a big chunk of data missing, a huge slice drop, something weird in the data, you better pick it out at the beginning than before you start processing and building your tractography and your connectome. So this takes time, I agree, especially if you have to do it 1,000 times. And so there are more and more softwares out there that can ease the process. But at the end of the day, a human has to do it. And I really believe that eventually we'll have smart AI systems that will be able to flag and reject the bad data sets for us in the future. Now we can start processing. Here's a list, and it can look quite scary because this is just diffusion pre-processing. You can see a list of 10 steps, okay, right now that are happening in the DWI community. Not all these steps are required. I'm just going to walk through the first 5 because they're all essential and they've been proven to be useful. And I think as a community now we agree they are. And the last 5 I'll keep for discussion later. I won't have time to cover them. The first thing is noise. Diffusion weighted images are quite noisy, but we have lots of them. We have the B0 image. We have all the different directions. Nearby directions and orientation space share a lot of structures together. So both spatially and angularly, these images share structure, even though they're quite noisy. And so now there's very good denoising algorithms that come from the image processing world that are very effective at removing noise, not blurring, so not affecting spatial extent and not blurring in the angular world. So I highlight a few tools out there that implement the data. Then comes eddy currents. Eddy currents and motion artifacts are very well established, and now we've been solving them for almost 20 years in the diffusion community. So if you don't correct them, this tends to lead to degenerate DTI tensors that lead to very sharp and rapid switching of on and off of the gradients. And there are very powerful software out there that are quite good at correcting eddy currents in motion. 2 of them are listed here, FSL and Tortoise. So again, yes, there is a consensus that this should always be done. One thing that people forget though, is if take into account these rotated B vectors for your analysis afterwards. I'll mention that later again, but if you're gonna be doing DTI and tractography and ODFs, you need to take this rotated B vector later. There are lots of challenges here again, you know, slice-wise motion, nonlinear motion, that these more advanced software now are trying to take into account. Then outliers. As soon as you start QCing your data in DWI, you often see these slices. These are called slice drops, or this I call the zipper artifact. It looks like a zipper in the images, or the ghosting artifact. Lots of these artifacts are corrected or can be accounted for. Again, because I said we have a lot of DWI images, a lot of directions, so we can easily robustify our estimators, use residuals, remove some parts of the image and interpolate with the other parts of the images. So one of my favorite tool to do this is the eddy command, which you need to activate the minus slice drop option. This is not done by default. So again, yes, here there is a consensus. This is important. Most software can handle this, but there are challenges. If there are too many slice drops, if it's a whole volume that's corrupted, do we remove it? Do we interpolate it? How do we deal with that? Imaging other organs like cardiac imaging or prostate imaging can be challenging for outlier solution, but overall in human brain mapping, this is well accepted and should be implemented. Last but not least is distortions. Again, you've all seen this if you looked at raw diffusion data. If you look at my cursor here, we have this large dip in signal because of susceptibility. As we're acquiring our images from the anterior part of the brain to the posterior part, and we're going from the sinuses and air into the tissue, we get this slice drop in our images, mostly an artifact, a geometric artifact. And again, this is an old artifact that people have tried to solve over the years with field mapping, with check and do QC of your DTI and ODF directions. Because even though your colored FA looks fine, right? This is red, blue, green, looks perfectly fine. Well, maybe underneath, you have this little sad smiley face. This is a flipped corpus callosum. This still looks fine in RGB. This is still red. This looks like a fine colored FA, but the underlying. So you should really make sure your corpus callosum, corticospinal tract, and in the other axis, your cingulum bundle, the splenium, the genio of the CC recommend using DICOM to Nifty X. To my knowledge, is the most robust multi-site converter of DICOMs to Nifty for diffusion. You rarely have issues of flipping and orientations and wrong affine matrices when you use DICOM to Nifty X. So I'm a bit solved problem. We can reconstruct crossing fiber information at every voxel of the brain. There's no reason to do DTI tracking anymore. Presentation. It doesn't have to be spherical deconvolution. It can be a multi-compartment, multi-tensor, multi-ball and stick modeling, but make sure you do have your crossing information and that you look at it before you do tractography. As a challenge or future question, there's still a lot of open question on how to do this well when you do group studies. How should we calibrate our response function when we do deconvolution? So I just want to throw it out here, and maybe that's going to raise question or discussion later, but this is still an open issue in large group studies. Should we fix it? Should we take the mean of the population? Should we just estimate it from the feet? We have our good local crossing fibers. Now let me give you a few precautions in tractography. First thing now that we accept as a community, now it's almost 8 to 6 years old, is that we should use what the acronym we like to call here now is ACT, anatomically constrained tractography. We no longer just use diffusion to do tractography. There was lots of years where we used to just threshold FA to get a good white matter mask and do tractography. This has been shown through the years to be suboptimal. We should really take good cortex parcellation. The only challenge here, of course, is pathology. If you throw in there brain tumors, lesions, and whatnot, keep that in mind. You anatomically constrained tractography may break because of bad T1 segmentation. But overall, in healthy human brain mapping, anatomically constrained tractography is now used, and it's a consensus. One thing now we can do better, and I've seen ET, where we can pimp up our tractography using the cortical mesh, just like we get it from FreeSurfer or from Civit or from a surface mesh analysis that we do use anyway for brain parcellation. Turns out it can be used to better seed and end tractography. So to the direction, using the normal direction to the surface to initiate tractography, to try to bring tractography towards the deep white matter, where anyway here the back project tractography. As tractography is having trouble reaching the cortex, we can help it project back to the correct white matter of more and more surface-based tractography in our pipelines. That will we really, today the topic is joining fMRI and diffusion, not really, but if you imagine joining fMRI, diffusion, EEG, MEG, brain parcellation, seems like the cortical mesh is really the perfect computational backbone to join all these modalities. And it really improves tractography as we reach cortex. Yet there are still issues with this. Here on the left, I show you an anatomically constrained tractography, the termination points. This is the cortex where you have a streamline finishing. So everywhere you see gray or dark, black, no streamlines have been able to reach cortex. So it's improved with surface enhanced tractography with SET, but there's still 30% of the cortex empty of streamlines, empty of tracks. And so this is kind of discouraging or a bit scary when you think about connectomics application. And this is known. Some regions are harder to track. Some regions are easier to track. And so it's much easier to get to the gyral crown than to the gyral blade or the sulcus. And so this is a very active field for tractographers to try to better seed and terminate and deal with these biases in tractograms. Because we know now that some regions are just harder to track. Just focus on this beautiful crossing fiber here. So I thought traverse this traffic or this complex region. So it's really hard for tractography to penetrate and traverse these walls of very strong highways of white matter. And so I love this figure here, where you see in color here, track density. So the easy part of the tracks are very dense, very visited by lots of streamlines. But as soon as you get into this two-way and three-way crossing, it's almost impossible for a motor track to traverse it. And we expect this to be full. We do have the motor homunculus here, and streamlines should fill up this space of these solutions. But I have to admit, at the moment, we don't have great solutions to eliminate false positives from tractography. So if you're doing connectomics, you need to keep that in mind as you're interpreting your results. So to conclude, really inspect your data. There are more and more nice quality assurance pipelines out there to generate automatically HTML reports, for instance, that you can go through them. So highly recommended. We saw the road towards consensus in terms of pre-processing. So denoising, eddy, outlier rejection, susceptibility, motion artifacts, and lots of open questions in the world of pre-processing, which are even more problematic as we're getting into high field MRI and 7 Tesla or connectome scanners of 3 Tesla. And in the end, be careful if you're pressing a button and reconstructing your connectome. Your connectome may be filled with easy to track tracks in preference, so be careful in your interpretation, and be careful if you're using pathological brains, because if you do have pathology, you have new obstacles, and probably your pipeline is not tuned properly, because 99% of the tools we develop are for healthy young adult brains and PhD students. So with that,
much appreciated. The Nature Comms paper is certainly one of my favorite in the diffusion literature, and I'd recommend anyone new to the field to check out that work on tractography. Perhaps we may leave questions to the extended discussion. So we have Maria Dibiase next. And then after that, we have a half an hour discussion section. If you have any questions, please go ahead and post them to the Q&A PhD at the University of Melbourne in our group in 2018. Her PhD was in diffusion MRI, particularly in applications in understanding pathology and schizophrenia. Upon completing her PhD, she took up postdoc work in Boston in the lab of Martha Shenton, and she was awarded an NHMRC investigator grant, which enabled her to return to Melbourne. And she's currently in my group at the Melbourne Neuropsychiatry Centre, and she's working on various projects, particularly diffusion MRI and linking diffusion MRI metrics with microstructure, as well as integrating genetic information with diffusion MRI data. So Maria, I'll pass over to you. Did you want to put up your slides?
Yes, sure. Thanks so much, Andrew, for that introduction. And I also want to thank Professor Maxime Decoteau for an amazing presentation as well. That was very informative. Today, I will speak more, as Andrew said, from a clinical applications perspective of diffusion MRI. And I'll begin by sharing a broad overview of the processing pipeline, including the, sorry, can you see my screen?
Yes, now we can't, but we did before.
Sorry, I couldn't see it on, sorry, I struggle sometimes with these.
We can see your desktop screen, Maria.
Oh, okay, and now you can see my presentation.
Yes, now we've got your presentation, but you can't see it, right? Is that the issue?
No, I can't see it, but that's okay. It's all right. So basically, we begin by, as Maxime mentioned, preparing the diffusion data, which typically involves quality control procedures and converting your data to your preferred format. And then we can pre-process the data to ameliorate artifacts. And then the processing phase involves more user decisions, such as selecting a diffusion model or processing technique, depending on your study question. And the last thing I've included here is result interpretation, because I personally feel this is a crucial component to any clinical diffusion imaging study. And so Max has already provided an overview of some of the key processing considerations for tractography. So here I hope to reinforce some of these points and to share some of the key challenges that I've faced in terms of processing and interpreting diffusion data. So I'll begin with a quick tip related to eddy susceptibility and motion correction, which I perform with FSL's eddy function. Now, version 6 and above allow you to generate several QC outputs. So one of the first questions I had when utilizing this function was, how best can I leverage these parameters to inform my decisions about retaining or discarding data? I do find it useful to plot these parameters for each subject to get an idea of the distribution and to identify outliers. So for example, here I show absolute movement, which measures motion with the B0 volume as the reference. And by doing this, I can detect those subjects who sit 3 or more standard deviations above the mean, which is indicated here with the red dash. And I can also represent this in a matrix format for later exclusion. But of course, we can deal with this information in different ways, and it may depend on the size of your data. For example, if you have sufficient power to remove these outliers, you can do so, but if not, maybe you want to take the conventional approach of just controlling for these parameters, or what I like to do is record the subjects with severe artifact and rerun my analyses without them. Now, these can be time-consuming steps, but they're very important, and here's one example of why, which is from Balman colleagues in 2018. If we just look here for a moment, this shows the impact of in-scanner head motion on structural connectivity, estimated here with a correlation between the streamline count and motion at each network edge. And it shows that about 12% of the connections shown here are significantly impacted by head motion. And this is amplified in the case of deterministic FA-weighted streamlines. And this makes sense, I guess, because we know that head motion can artificially inflate FA estimates in low anisotropy regions, and it can diminish FA estimates in highly coherent white matter regions. These are just some key references that discuss these preprocessing artifacts and some of the sources of artifact on the resulting diffusion measures. I'll now come down here to the issue of harmonization, which may or may not be a matter of concern for your data. But basically, you'll know upon checking the raw DICOM data. And this is the first preparation step for your diffusion data. So here's an example portion of data which shows the subject ID in the first column, and then I've just extracted several key acquisition parameters, including the scanner serial number and software ID. Now for these data, I was told that the same scanner and software were used across the entire data set, which is clearly not the case. Multiple scanners have been used, and for this particular scanner, this subject was scanned using a different software. So it is really important to check your DICOM data, as Maxime was saying. So for these data, I actually harmonized them, in this case with a spherical harmonics procedure. So just briefly to do this, we select a reference and target site with about 15 to 20 healthy controls. Then we can create several what are called rotation invariant spherical harmonic features at each voxel, and these just capture different components of the diffusion signal. We can create templates based on these features and then learn the differences between the reference and the target site to produce scale maps, which are then used to update the signals from the data in the remaining in that site. Now, of course, there are different harmonization protocols that we can use, including statistical harmonization, and these are compared in Pinto's 2018, no, sorry, that was from this year, Pinto's review from this year in 2020. Now moving on to the processing phase, and one of the key challenges here is measure selection. Now, this is not an exhaustive list of diffusion models, but as you can see, there are a range of models that we can apply to the data to generate several diffusion measures. And the decision to select one will obviously be driven by your study question. So for example, I'm interested in understanding the biological basis of psychiatric disorders. So I often turn to these more biophysical models so that I can make inferences about specific tissue properties. For example, free water imaging allows you to separate diffusion in the vicinity of the tissue from freely diffusing water molecules in the extracellular spaces. Now, the other important question to guide measurement selection is whether or not your acquisition was appropriate to fit a particular model, which is then used to estimate diffusion measures. So as shown here, typically lower B values are better for tensor fitting. And then the more parameters that you estimate, or the more refined a model is, the more difficult the estimation process, and the more data is typically needed to estimate these parameters reliably. Another key decision along the processing pipeline is the particular processing technique that you choose to summarize your diffusion measure. These methods can be viewed in a timeline in a way because they grow in popularity from A to D. And the region of interest analyses just use a white matter atlas so that you can compute an average diffusion measure from voxels within a particular white matter region. And then the track-based spatial statistics, or TBSS, allow for voxel-wise comparisons across each voxel comprising this white matter skeleton. So this is really getting at the core white matter. Tractography we heard about, and then the connectivity measures allow you to analyze the brain from a network perspective. If we just follow on from Max's talk, of course, one of the key advantages of tractography is that we can reconstruct fiber trajectories in the brain. But one of the key disadvantages is that quality control procedures can be time consuming, particularly for larger data sets. But from my experience, viewing and interpreting probabilistic tractograms can be challenging, particularly in terms of judging the location of true anatomical boundaries, which can again be obscured by false positive streamlines. And interpreting the raw streamline count can be challenging in terms of the biological relevance as well. The last point that I'd like to cover is relevant to result interpretation. So we're often warned to please interpret our results with caution, but what exactly does this mean, and what factors should we consider when interpreting and reporting our results? When I was putting this slide together, I was reminded of this quote by Derek Jones in 2004, which stated that diffusion MRI measures just one thing, the dephasing of proton spins in the presence of a spatially varying magnetic field. Now, this is true, but in practice, this information may not be very interesting to a clinical readership. So I'll quickly present 4 quick questions that I use to guide interpretations of my findings. One question that I ask myself is, was the acquisition optimized for the model? And there are now numerous studies that address this issue for different models, and that can be referred to guide considerations on the matter of acquisition. Yeah, I just wanted to raise that, of course, there may be times when your acquisition wasn't the most ideal one to fit a particular model. So here's an example of how I addressed this in a recent paper that examined the link between brain free water and peripheral inflammation. So in this case, I used free water imaging with single shell data, but the, I guess, ideal case would be multi-shell data. And I've just stated here that single B-shell data available here. With these data, the free water imaging relies on spatial regularization and may be biased by T2 effects, as well as perfusion and pseudo-diffusion effects from blood. I think this is okay as long as we're transparent about the limitations in acquisition. The second question that I ask is, what are the model limitations? So if I've used diffusion tensor imaging, of course, a major limitation is that it assumes that water molecule movement is free or unrestricted, which results in this Gaussian distribution of water molecule displacement. Now, this might be the case in free water, like in CSM, but it's an unrealistic assumption in the case of tissue or in complex white matter structures where diffusion will be constrained by cellular membranes and organelles within cells. So in this case of tissue, the, I guess, water molecule movement will be more restricted, and this distribution looks more elongated and no longer Gaussian. So we can address this by applying a more advanced diffusion model like diffusion kurtosis imaging, which provides measures of microstructural restriction. But even in these more advanced models, there are assumptions that need to be considered in interpreting your data. A third question relates to the potential sources of non-biological variation. And here are just several examples, including motion, gradient nonlinearities, eddy current susceptibility, Reichian and non-central chi noise, and processing artifacts. And the fourth and last consideration that I'll mention relates to biological interpretations. And I want to briefly draw an example from the tensor metrics axial and radial diffusivity, which became very popular after 4 seminal studies in mice. In 2002, Song and colleagues found that radial diffusivity was significantly increased, but axial diffusivity was not altered in congenitally dysmyelinated Shivera mice as compared to wild-type mice. And then in 2003, Song and colleagues found different patterns of axial and radial diffusivity during the course of optic nerve degeneration in a mouse model of ischemia. So for example, they showed that 3 days after ischemia, the axial diffusivity was decreased, which was consistent with these histological findings of axonal degeneration at this time, but no detectable demyelination. And there were a couple of more studies by Song and Sun, which in general found that if you destroy axons, the axial diffusivity goes down, and if you destroy myelin, then the radial diffusivity goes up. And this is very intuitive, but an important point needs to be made that it doesn't mean that the converse is true. So if radial diffusivity is high, then myelin is destroyed. Of course, this isn't necessarily the case. There are many biological phenomena that can change your signal. So this would be a fallacy in logic. If we go back to the same study that I just mentioned before, this is just an example of how I formed a biological interpretation of diffusion data. So here I just started explaining what we measured in concrete, though lay terms, because this was geared toward a clinical readership. Then we stated the biological hypothesis. So in this case, this was that inflammatory processes are hypothesized to contribute to a free water increase. And finally, we pull in evidence, in this case from animal and post-mortem work, to substantiate this biological claim. Now, of course, in the discussion section, again, we mentioned limitations and other sources of variance of this measure. Okay, I'll end there. I just want to thank the people who have taught me about DWI, including my supervisor, Andrew Selesky, my former supervisor, Ofer Pasternak, and Yogesh Rathi, who taught me quite a bit about harmonization. And I'm happy to provide this link in the chat if anyone's interested in downloading the slides. Thanks very much.
Thanks, Maria. That was great, really jam-packed with lots of useful advice for upcomers in the diffusion MRI field. So we have an extended discussion session now. We've got probably 15 or 20 minutes. We've got some questions. Max is ahead of the game and has already answered some of the Q&As, but I encourage, if anyone has any questions or feedback or remark, please post them on the Q&A session, and we can hopefully address those now. Perhaps I'll start with a question or a comment, and I guess this is both to Max and Maria. both of you have highlighted the importance of eyeballing data as well as quantifying the extent of motion. Of course, Max, as you pointed out, this can be an arduous and time-consuming task. So as data sets begin to grow, so if we start looking at, for example, the HCP, or where we're in the thousands, but if we go then to the UK Biobank when we're in the tens of thousands, how do we go about doing this? How can we perform these quality checks? Surely we need automated pipelines. And what would be your recommendations in terms of these kind of quality checks? Max, maybe did you want to start?
Yeah, I'm thinking about this every day because we're starting to consider the UK Biobank, and it's a nightmare. It's not possible to manually QC all of this. And so recently, GB Pauline, I don't know if you know, he's in the OHBM community, and he's a very, intense neuroinformatics big data guy. And he always tells me the opposite. He's like, just process your data and look at the outliers at the end.
Yeah.
Which is one way to go. But I'm kind of scared to do that with diffusion, with, our 30 plus different parameters in the pipeline. And so I would tend to recommend maybe a halfway solution where you do process, but you look at outliers in each of the separate big blocks of processing. So for instance, you're talking about motion, and Maria told us about the Eddy QC tool. But so if you flag that, for instance, somebody has had motion beyond X degrees, well, you can try to automatically put aside patients with high residual, large motion, big distortions, and try to prepare a database of people you should QC, and the others you can leave aside. And like I said, I really truly think AI will help us here soon. And again, AI will probably qualify for us data sets to look at. So if you have 20,000 or X thousand data sets, if you just have to QC 10% of them, well, that's already 90% where you can just trust the AI system. So I think this is coming, but nothing to my knowledge yet to do this.
Thanks, Matt. Maria, did you have anything that you...
I definitely agree. So this is what I tend to do, is just run and generate QC outputs, set those aside with severe artifact, and go back and look what might be happening in these particular data, or exclude them in the analyses. But I think it gets more complicated with things like tractography. So one of the things that I like to do for even just the outliers, usually for the whole data set, is, for example, use something like white matter query language, where we can automatically label tracks. So for example, we can extract the uncinate fasciculus for all the subjects and have a look at cross-sectional snapshots of these fibers to get an impression of whether the fibers are curving in the right direction, as we would expect, a C-shape direction, and whether there are actually streamlines present. So if there aren't, of course, and we can quantify this, we can go back through the processing pipeline and see what might have happened with this subject.
Thanks, Maria. I think that addresses the question. Perhaps we'll move on, and I guess this is a follow-on to a certain extent. So this is a question from one of the audience members. It's Maria Dibiase, and the question is for someone that's starting off or beginning a diffusion MRI study. I presume the questioner is asking about collecting data. What would be some of the key recommendations in planning a new acquisition or acquiring new diffusion-weighted data? Max or Maria, would you have any specific, maybe some key guidelines in terms of the acquisition scheme, and what would be recommended in terms of perhaps number of shells, gradient directions, B values, and so on?
I think it depends on how much money you kind of have to work with in terms of the acquisition. But please, I think, Maxime, you'd be better at providing information on this matter.
Yeah, well, first thing I would highly recommend, and people rarely do this, but do a little pilot study. Just you, yourself, a couple students, go in the scanner and scan yourself 2 or 3 times before you even start recruiting patients. And then You know, make sure your pipeline works and address variability. We never know actually what's the variability of our measurements. So, you know, if you're interested about FA or DTI, well, do it. And then really talk to your scientists or MR specialists to try to develop an acquisition scheme that fits your needs. I see too many people being a bit of fashion victims of the field and just wanting to acquire more and more and multi-shell and naughty and B-tensor encoding stuff without really knowing why. And so you don't need all that data probably. And acquiring all that data means you're not going to be able to acquire more, or, you know, so think about study designs. So for instance, if you're just interested about DTI, I would almost recommend you a 30 direction protocol, but do it twice maybe. Do it at the beginning of your protocol, do your fMRI, your T1, your flare, and finish with another DTI. And then you can convince the reviewers that all your results are reproducible with test retest, for instance. But this is the computer science in me talking about, you know, knowing all these issues. Because for me, I always wonder, is it the pathology driving my result, or it's my bad methods, you know? And so just make sure it's not your methods that are driving your results. Make sure it's really your research question and your population. So that's my a bit pessimistic or my advice, at least when you start, you do have control.
Good advice. So maybe we'll move on to some of, we've got a fair few technical questions here. I think, Max, this one is directed to you. So in your presentation, you alluded to the bias of many streamlines, or there was regions on the cortex where there wasn't any streamlines that terminated. And this was raised as an issue. And I think this is more generally what you're referring to here is the so-called gyral bias, which was pointed out 3 or 4 years ago in a couple of really nice papers. And so the question in here is really asking, are there any methods that can be done to deal with this gyral bias? And perhaps if we take this question more generally, what are some of the other biases in tractography that we would need to be cautious of? And how would you suggest that newcomers to the field would overcome, or what are the best practices to overcome or limit the impact of these biases?
Yeah. So great question. It's a tough one because I don't think all the tools are ready yet for mainstream usage. So that's the issue. You almost need to collaborate with my lab, or, you know, I know MR-Trix is developing the mesh-based ACT or FSL. So the first answer is for gyral bias. I really think we have a better handle on it using cortical meshes. So I don't want to say it's a solved problem because it's not. But we have a way towards minimizing it. So that's the first thing. But what people forget is beyond the gyral bias. What I was trying to show you in between these 2- and 3-way crossing, I mean, some tracks are just harder to reach, harder to track. They go through little thin bands of white matter, let's say around the, you know, the insula. So I always say we have a huge challenge tracking around deep structures as well, around the thalamus, around the putamen, around the insula, where everything is so packly dense, everything looks like a bottleneck, and you don't know where to go. Tractography is really limited there. And so what do we see in tractography? Well, we see the easy big stuff, mostly. And the small stuff is easier to miss or harder to track. So just for instance, you know, the most popular memory track is the fornix. Well, I give you all a challenge to find the fornix in your data. Most of you will not find it. You will have to, you know, tweak tractography to get it. So essentially what that means to me is that we need smarter tractography. We need tractography that adapts to the different locations in the brain to be able to track better in harder and thinner regions. And this is wide open to, I think, this is really wide open. So, So
and
again,
what about acquisitions, Max? I mean, is there scope there for improving diffusion-weighted acquisitions to make headway in this area, or is it really just on the algorithmic issues of tractography?
Oh, for sure. I think we were a kind of bit fashion victim towards high resolution, high angular resolution. You know, more shells, more B values, more directions. But I think we should have focused on more spatial resolution as well. So this comes a little bit with 7 Tesla and, you know, millimeter isotropic acquisitions. But if we want a revolution in tractography, we will need higher resolution data.
Yeah.
To get through these harder to track regions, I think. So, you know, the physicists out there, there's definitely lots of room to probably have less orientations. Like I go crazy when I see these protocols with 500 orientations. I don't think we need that.
Yeah.
We should do smaller voxels and average to get
build
better
up our SNR.
Because I know this. I've done it in my PhD. Like the orientation resolution plateaus. As soon as you have 100 orientations or more, you're kind of plateauing.
Yeah.
So it's a waste of time to acquire more than that.
Yeah, point taken. I think I'd like to step back now a bit, and I think there's many of us in the audience that are working on or using diffusion-weighted imaging to answer basic neuroscience questions about brain anatomy and brain connectivity, as well as clinical applications. And so the question is really about the biological validity of diffusion MRI, particularly things such as streamline counts and our diffusion metrics. So if we turn to streamline counts, I mean, streamline counts were used to measure structural connectivity in the very first connectomes by Sporns and Hagman, and they pretty much remain the default measure of structural connectivity. So I wonder, Max and Maria, if you had any thoughts on what are we actually measuring with streamline counts, and are they a biologically valid measure of structural connectivity? And do you have any broader comments on understanding or interpreting not only streamline counts but other voxel-wise measures such as fractional anisotropy from diffusion MRI? Perhaps we'll start with you, Maria. I mean, you made the important point that we're actually measuring spins, and so when we use these spins to actually infer things about brain disorders, there is a big link there from our inference to what we're actually measuring. Maria, do you have any thoughts on?
Yeah, sure. So I think it's a really important consideration, first of all, and Fernando Calamanta actually spoke about this in a recent paper. When you consider probabilistic tractography, every time you run the algorithm, you're producing different tractograms, he noted. So of course the reproducibility is low. And then you have the impact of bias and error in reconstructing streamlines, and therefore the streamline count won't necessarily reflect the underlying density of fiber bundles traversing a voxel. So I think that there have been some methods like SIFT and filtering approaches that remove some of this error and increase the biological meaningfulness of things like streamline count. But we, I guess, don't know whether to apply this in the case of brain disorders because we may be removing some of the real biological variants that we're interested in. I generally think that it can be used as a proxy for connectivity strength, and also we can combine it with other measures of the streamline like the cross-sectional area or the volume, which may give us a better picture of the biology of connectivity strength. But of course there are factors that we need to consider. I think Maxime raised one. Of course, with bigger brain regions, streamlines are more likely to terminate in these bigger brain regions as well, so we need to address these before we're inferring about the biology of streamline count.
Max, what are your thoughts? I mean, in terms of streamline counts, what are we really measuring there, and how do we interpret these in a biological or in a perhaps a clinical context? How would you recommend that we go about this?
It's a tough one. And I mean, to me, there's nothing biological about streamline count. So that's said. Having said this, it's data driven. So that's true. It's very good at, you know, building a data representation of my brain. And for instance, if I did this test many times, if I build my connectome and I have all my students connectome in the lab, I go back in the MRI, I find myself very easily. My connectome is my connectome. It's different from my student's connectome. So what I mean by that is that even though this streamline count may be very far from the biology, it's still representative of my geometry and my diffusion weighted image or snapshot of my brain. So there's something powerful in it. I think where it becomes dangerous is the interpretation, because to me, as a developer, as a computer scientist, to me, it's just reflecting easiness to track.
I think
the more tracks you have between region A and region B, to me, it's just telling me it's easier to go from A to B. There might be biological reasons for this and obstacles and different, but to me, there's less obstacles for sure. Yeah. There's less crossing fibers, there's less obstacles. What does that tell us about axon biology? I think it's more about the geometry and the easiness to travel down a road and reach a specific area.
Yeah. I mean, I agree from a technical perspective, that's by definition what we're measuring. I wonder though, if we do want to really see the clinical utility or neuroscience utility, do we need to, so when we see an association between streamline counts and, for example, treatment outcome, or streamline counts in response to, for example, deep brain stimulation, is there a need to interpret this further, or is there any scope that we can make advances to help us unpack these associations that we see with treatment outcome or with a certain fMRI, for example? So if we correlate fMRI connectivity, we'll probably see that in the next session with structure, we see these associations. And I wonder if that would provide us with some hints as to what we're measuring with structural connectivity. I know this is a difficult question, so I don't know if there's any answer, but for any concrete answer, but Maria or Max?
We're close to being able to do better.
Yeah.
Very close. Not yet. And it's in part our fault, the methods developer. We haven't provided you guys better tools for years. Yeah. And so I think it's fine to use it as is, but you know, for instance, Andrew, would you, if a drug, a pharmaceutical agent was increased after using this drug, you would see increase in track count or reduction in track count. Would it be safe for you to kind of bring this to the FDA to say, look, my drug is working. I'm regenerating axons, or because that's what we want, right? That's kind of what we want.
But of course, I mean, we can't claim that. We can't claim that we're regenerating axons. I think that's, I mean, from my opinion, that's one of the challenges of the field, is really making that and advancing in that direction.
An important
sorry, Maria, go ahead.
Oh, no, go ahead. I just thought that an important point to make here is just because something's not biologically valid doesn't mean it wouldn't be clinically useful in terms of prediction. So we can maybe predict treatment response, which would be handy to know in a lot of pathological conditions. Even though we may not be able to treat it based on the streamline count, maybe it is okay if we're just basing our predictions on these geometry and diffusion factors.
Yeah, I think that's a good point, Marie. Perhaps it's not so much about understanding or interpretation, but if we have a black box of prediction where we can put in scans and we can get an accurate out-of-sample prediction, perhaps there is clinical utility in predicting response and so on. Marie, I think this question might be for you. It's another clinical question from an anonymous attendee, and it's specifically about choosing templates for, I assume, registering data in clinical populations. And so given that we want to be sensitive to take into account individual variability, do you have any advice on choosing a registration template for group analysis? I think this question could probably also be interpreted a bit broader in terms of what are some of the challenges that we might need to face when doing group analysis and accounting for differences in brain connectivity and brain structure across individuals.
I do think it's very method dependent. For example, when we use something like the skeleton, there can be misalignment errors. I mean, Andrew, you had a paper on this. If we're projecting voxels onto a skeleton, there may be misalignment that might cause error in our data and sources of artifact. In terms of registering to a particular template, I think it depends on the age of your sample. There are different templates across different ages, and it also depends on what you're comfortable with. I think in a way, I always turn to the same templates, the fMRI or the Enigma template, because I find they're very robust and they've got good anatomical information that we can register to.
Thanks, Maria. Max, did you have anything that you wanted to add, or should we move on?
I don't know if Maria agrees with that, but in some of the projects I've had recently, I prefer to build my own template, actually. So these tools are more widely available now, and I use ANTS for this, ANTS. And if you have a large enough population, let's say I had an Alzheimer's group and I wanted everybody in common space and MNI space. Well, the first thing I did is I built an FA and T1 template of my group. So these are all people from Canada, 60 plus years old, and I didn't want to register them. I didn't want to choose a template because I didn't know better, and I don't have enough experience with that. So it took, a week of computation, but I built my own template, and then I used it for the whole project.
I think it's always more accurate to build your own template. Sorry, I misinterpreted the question there. I thought you were talking about already available templates. Yes, even with voxel-wise analyses and things like this, it's better to build your own. I think you come into difficulty when we're doing more longitudinal work where we add people later on. So I've always struggled. I'd like to get your opinion, Maxime. In this case, would you build your own template again after recruiting more individuals, or?
I would probably stick to just out of laziness as well. If I'm happy with the version 1 of the template, I would probably stick to it.
Yeah.
But I've never thought hard about this. I've never had this problem in my career.
So we might move on. We're rapidly running out of time, and I think a good lead into the next session is that we have a question once again from Miriam about integrating. And I think, Max, this is something that you brought up in your wonderful presentation as well, about integrating functional MRI and diffusion weighted measures. And so the question is really, what are some of the considerations in integrating diffusion MRI metrics with other modalities, namely functional MRI and functional connectivity? Max or Maria, do you have any thoughts on or recommendations in terms of multimodal integration of diffusion MRI?
I love multimodal work. Yeah, I think it's beautiful. I think here there's a trade-off between, having these multimodal, beautiful studies, and even you can concurrently measure. I think Maxime's done a bit of this, concurrently measure PET radioligands to try to understand the mechanisms underlying the large-scale MRI signals. Of course, then maybe your sample size would be reduced because they're just more expensive to do. But again, this highlights the point of registration. We need to register probably to the T1 structural data so that we have all of the multi-modalities aligned to proper anatomical information for this individual.
What about in terms of validation? So if we can show that for example, if we have a structural network from diffusion MRI functional connectivity, and we see that they recapitulate certain network features of connectivity, can we use other modalities such as fMRI to, in a sense, validate or give confidence in the connectomes that we're mapping with diffusion MRI? Max, would that be something that you would recommend?
I think you're more an expert than I am on this. I read a thesis recently from your group that did a pretty good job at trying to do this, and I took some notes down because I think it's a smart way to start complementing each modality and probably not validating in a strong sense, but confirming things with a separate modality. So I do like these ideas, and I do think, again, computationally, going through the cortical surface is probably a smart way to bridge the gap between all these modalities once they're registered in T1 space, for instance. So, and also start using the power of streamlines or tracks to bring in, to project to the cortex whatever feature of the white matter you're interested in. Because anything is possible then. You can map myelin, mean FA, track count, whatever. And that's really, really going towards real, pure, exciting multimodality work to me.
Yeah, no, I agree. So perhaps the last question, we're running out of time, it's from Marcus Gray. It's a very brief one. Any thoughts on the utility of intra-axonal diffusivity measures? Maria or Max, any quick responses on the validity or utility of intra-axonal measures?
Go on, Max. I know you've done more on this pixel-based stuff.
I haven't done much more, but I really I'm a believer of it, and I'm having ideas into mapping EEG activity along streamlines and try to either influence the inverse source localization problem in EEG using connectivity, but also vice versa. Use time delays and connectivity, and maybe eventually microstructure or G ratio type conductance physics to try to explain the delays in EEG signals using connectivity. So I think this is a really hot topic. If I were as a PhD again, I'd be keen on looking into this.
Thanks, Max. Maria, did you have any final? Should we?
No, this is something I haven't done very much. I do find it very exciting as well.
Yeah. So I think we might need to wrap it up. We're right on time now. I would just like to once again thanks Max for joining us this morning or nighttime over there in Quebec for a really wonderful talk and wonderful discussion. Max really is a world-renowned expert in the field of diffusion MRI. So it's been a pleasure to have him present at our webinar this morning. I'd also like to thank Maria Dibiase for giving a wonderful talk on pre-processing and also participating in our discussion, and for providing some wonderful insight on her work and interpreting diffusion measures. So I'll hand off to Adil, who will moderate and chair the next session on functional MRI and pre-processing strategies for that modality. So thanks again, Max, and thanks again, Maria.
Pleasure. Thanks for having us.
Thanks, Maxime.
Thanks a lot, Andrew. So that was a wonderful session. There was a lot to learn from that. And there were quite a few common themes that we will probably see emerging in terms of QCing data and noise correction. So thanks a lot. So I'm Adil Razi. I'll be chairing and moderating this session. We have two brilliant scientists who have joined us. We have Cathy Chang and Sasuke Balman. So we will start with Cathy. So Cathy has done pioneering work actually in terms of characterizing dynamics of functional connectivity. She's an assistant professor in the Department of Electrical Engineering and Computer Science at Vanderbilt University. Before joining Vanderbilt, she was a postdoc at the National Institute of Health and received her PhD from Stanford. Her lab seeks to advance understanding of human brain function through techniques for analyzing and interpreting neuroimaging data. So over to you, Cathy. And yeah, looking forward.
Thank you so much for the intro. So let me try to share my screen here. All right, does that come through okay?
Yes, definitely.
Okay, great. So resting state fMRI, like DTI, is a very large area. perhaps more questions than answers, but in the next 20 minutes, I'll hope to raise some of the main issues. Oops. There we go. So I wanted to start by discussing a bit about why we have issues in resting state processing in the first place. So what makes this tricky? And I think that one major reason is that our data are mixtures of, oh no, why is this not advancing? Oops. Are mixtures of neural and non-neural signals, and at a lot of the confounds can be larger than the signals that we may be interested in. And that's not necessarily a bad thing, except for that these tend to be difficult to disentangle. There can be complex interactions and covariations between them. Another thing is that, sorry, I'm not sure why this is that it's often hard to tell if a particular processing step or pipeline is doing the right thing, because we may not know what our resting state data in a given subject should look like. So we have to come up with heuristics to evaluate our methods. Related to that, we don't have an explicit model of resting state time courses, so we don't have the option to do things like we can in tasks, like include more trials in order to better isolate effects of interest. So for resting state, a lot of the challenge is trying to separate the parts of the signal we're interested in from parts that we may not be, and when we go about processing our data. And so just as a kind of general overview and context, here is one typical resting state processing pipeline. This is just one example. There are variations, of course. And I've roughly grouped these into steps that you might do in the native acquisition space. And here are steps that would involve spatially warping your data into standard space, for example, if that's of interest. And then trying to remove structured temporal confounds. This is not a perfectly linear thing, of course, like retro-ICOR, for example, is also a method for removing structured confounds, but that needs to happen in the native space, whereas certain denoising steps may happen before or after the alignment to standard space. And so I just included a couple of notes here. One is that I wanted to emphasize here too that it's really important to look at your data, not just at the beginning, but throughout the output of various processing steps as well. And there's been a lot of development of different QA pipelines that can be helpful for that in the fMRI field. Another note is that for some steps, there's a clear right or wrong order to doing them, but in other cases, the optimal ordering is not clear. For example, these steps interact with each other, and as well as with how the data were acquired, such as whether you got your slices in an interleaved or sequential fashion. There are many choices at all these steps. I won't have a chance to go into all of them during this talk, but happy to return to them during this discussion. Here, I'm mainly going to spend most of the time in this area of reducing confounds. And one reason is because this is a place where I think the practices in task and resting state fMRI diverge. And another is that there are a number of issues that come up when deciding amongst the various possibilities that we have for reducing confounds. And so here is a partial list of some of the major steps that we may consider. Generally, there are some that build specific sets of temporal regressors or apply temporal filtering, and others that are more multivariate data-driven. And so I guess we're going to go into some of these, but as a few kind of overall notes, in the resting state literature, it can be hard to synthesize and get a clear picture of what are the kind of clear findings in the field, because different studies use different steps or different combinations of these different steps. And a lot of these may come with different parameters and thresholds to choose. So there's really a large space of possibilities in how we can process our data. And then we always worry about removing too many things from our data. So we wouldn't want to go and apply all of these, because the more components or the more regressors that we toss out, then the more we sacrifice the degrees of freedom in our data and potentially remove things of interest. And then as for which steps to apply, there are a lot of questions there, and there may not be one single optimal solution. In fact, even whether we treat one particular part of our signal as signal or noise may depend on what we're actually trying to study. And so I think that in order to try and make good choices for our particular analysis, that goes very closely with trying to understand what sources of fluctuation are in our data, what are their specific spatial and temporal properties, and how they interact with the effects that we're trying to study. So these are the areas I'm going to focus on. Well, I'm going to give a quick tour through these, some of the main sources of resting state variance. And for some of these, I'll maybe be able to offer some recommendations, but for others, I'll mainly just raise some issues and hope we can have some more discussion about that afterwards. So one of the major concerns for resting state has been head motion. It was pointed out by these and other papers that movement of the head can systematically alter patterns of correlation in our data. And so what this does is produce displacement of our images, which we may address by spatial co-registration. But even after that, we're left with residual intensity fluctuations, for example, due to movement in the slice direction, causing slices to no longer see the RF excitation pulses at the expected times. And so I think it's agreed upon that we should do some sort of co-registration, spatially aligning our images to handle motion. But in terms of removing these additional effects, we have many options. So we could regress out motion parameters, or their squares or their derivatives, and the number of things that we regress out. I mean, that's kind of a choice we have to make in our study, kind of open question. Another approach is to censor or downweight high motion timeframes. And in some cases, this has been shown to be more effective than regressing things out. But then we also have to choose some kind of threshold for doing that. And then we have some state-of-the-art multivariate approaches which have been developed, which are trying to basically unmix motion and from other types of artifacts, from signals that may be neural based on their spatial and temporal signatures. So these are all retrospective corrections. They can be applied after the data are collected. There are also prospective approaches that can be helpful to minimize those effects in the first place, such as adjusting the acquisition based on real-time tracking of head motion. I think that, if possible, it's best to just try to take measures to prevent motion in the first place. This is a head kind of mold restraint that's been recently developed. You may not need something, or sometimes it's hard to get one of these or to get your subject to actually wear it. There may be like less invasive solutions like kind of a tape or something you can put on their head, just a little bit of feedback. Of course, easier said than done with some populations, like small children. So aside from motion, we also have to worry about other physiological effects. And so another source, as you may see here from this high TR data, are pulsations of the brain from cardiac activity. And we also get effects that are synchronized with breathing cycles. And so these effects are happening at frequencies typically higher than what we study in our conventional resting state studies. So like respiration is around 0.3-ish hertz, cardiac around 1 hertz. And this frequency scale, I mean, where it's often typical to bandpass is like around like 0.1 or so hertz, which is like way down here. So we may think that, is it possible to just apply a bandpass filter to kind of reduce these kinds of effects? But the problem is that with most TRs, these peaks are actually going to alias into our lower frequency bands, meaning that even if we have like a fast cardiac cycle, it's like at 1 hertz, if our sampling rate is longer than the Nyquist rate, so then we're actually going to be undersampling that waveform, and our peak is going to appear as if it's at lower frequencies. And it can actually directly overlap with our spectrum of interesting bold signal. And so you kind of figure out where it might be based on the person's heart rate, or as well as the TR that you're acquiring the data. But unless you've acquired the data at sufficiently small TR, then the frequency domain bandpass filtering won't really help much there. And then another thing to be aware of is that even if your TR is small enough to avoid aliasing the main fundamental frequencies, you may still be aliasing some of the higher harmonics, which are smaller, but could also contribute non-trivial variance to our data. So here's a spatial map of the distribution of respiratory and cardiac noise for the fundamental frequencies, as well as for the second order harmonics. And one thing to note here is that there's a lot of variance explained in areas like, especially like in the insula or in the brainstem. And so correcting these effects may be particularly important in such regions. So in terms of how we correct for effects that are aliased, if you've collected respiration and heart rate data, like with respiration belts or a pulse ox signal, then we can try to build regressors that capture this effect. And so retro-I-core is one such approach that's based on modeling our fMRI signal deflections at every heartbeat or every breath with a basis set of low order sinusoids. And so what this is going to result in is a set of regressors that are essentially composed of undersampled sinusoids sampled at the precise places where you've acquired a given fMRI slice relative to the cardiac or respiratory cycle. This is actually a pretty old method, but still one of the main tools used for today for correcting these effects, at least from a model-based perspective. Although in practice, it's not that widely used in fMRI because it does require that we gather physiological recordings, which is not always done. Although I do always recommend collecting heart, cardiac or respiratory data when possible, because it's usually not that hard and it can give us really valuable information that can help us interpret our data. But in the absence of these recordings, spatial ICA-based methods may be effective in removing these cyclic physiological noise, as well as motion effects. And so some of the prominent methods that are being used now, FIX is one. It's an ICA-based approach. And multi-echo ICA is based on the property that if you happen to have collected your data with multiple echo times, then real bold effects like you may expect from brain networks, their percent signal change will scale up as a function of echo time, whereas non-bold effects will not. And so at the end of the day, you may be able to rank your components by the degree to which they show an echo time dependence, and hopefully get something like artifactual components ending up at one end of the spectrum compared to resting state networks. Personally, I find this approach to be very effective, but it does require that you have collected multi-echo ICA or fMRI data in the first place. So retro-I-core and spatial ICA-based methods can be effective at removing some kinds of physiological effects like the cyclic noise we just discussed. But there are other kinds of physiological effects that can be harder to pull out with these kinds of methods. And one relates to slow natural changes in the amplitude of your breathing over time. So essentially the amplitude of the respiration waveform rather than the individual breaths themselves. And so to illustrate the effect on fMRI data, you might make some carpet plot like this where you've lined up your voxels, and you can see that the major modulations in the respiratory waveform are linked with these large deflections in the signal, and also show up in this fMRI whole brain average. And there's a few reasons why RVT is our respiration variation effect is especially tricky to separate from our neural signals. One is because these are actually low frequency fluctuations, so they're directly in the range of our hemodynamic signals. It's not just because of aliasing, so sampling at different rates is not going to help. And it also correlates widely with voxels across the brain. And so there's a spatiotemporal overlap with neurally generated bold signals. And in fact, this is also a bold effect since it comes from modulation of cerebral blood flow and the vascular tone under control of the autonomic nervous system. And changes in heart rate also show a similar effect. So just to illustrate that these effects may persist after some common processing steps, this data had undergone fixed ICA, and I took a subject and just map out the percent temporal variance explained by these low frequency physiological effects, where you can still see there's a pretty substantial portion. And then to illustrate this in connectivity matrix form, we might take the same data set and make an ROI to ROI correlation matrix, and then compare what it looks like before versus after regressing out respiratory and heart rate related effects. And so how do we model those low frequency signals? One way that's prominent in the field is to create temporal regressors that try to capture these effects. And we can try to obtain these by monitoring the respiration and heart rate during the scan and trying to then fit models in terms of how these propagate into the fMRI data. And so one method is by trying to estimate response functions. So respiration and cardiac response functions, just like similar in spirit to how we have like a hemodynamic response function that we can involve our task design with to get a regressor that basically is a time course that models what we think the fMRI signal changes due to RVT and heart rate are. And those may be used as nuisance regressors projected out of the data to try to reduce these effects. Now, interestingly, it may be important to fit different models over different parts of the brain. And in this study, we actually we're looking at that specifically and finding that different parts of the brain seem to have different physiological response functions, which may not be so surprising. But one interesting thing I thought was that if we cluster the brain areas that have similar physiological dynamics, they tended to form very organized spatial patterns that look a lot like resting state networks. And the kind of spatial overlap between physiology and neural networks was also shown in this recent study from Molly Bright, where what they did was they presented a block design task, and in the same scan, they had a CO2 vascular challenge. And so if you do an ICA on that data, what they end up finding is a kind of paired components where you get some network that looks like default mode network, but you may also get another network that looks like default mode network, but its time course actually is closer to what's going on in the vascular stimulus. And so I think that, and then, you know, the idea wasn't to say that like all of the neural networks are just vascular, but that it can actually be tricky to tell which, you know, if you're to run an ICA, which one is which. So I guess one thing that suggests is that it is useful to collect physiological data when you can. And another thing is that there may actually be an interesting kind of spatial alignment between these kinds of networks that may be beneficial for like how our brain functions. Another consideration, I guess, in whenever we try to model and remove effects like this is that there can definitely be covariation between physiological and neural effects. So this figure is from a really nice review article from Tom Liu. I'd highly recommend it. And it illustrates the close interconnection between effects that we often take out, like head motion or physio, and brain activity, and reminds us that those effects are also under control of the brain. And so we should just think hard about what is signal and what is noise, especially if we're trying to study something like autonomic function or, you know, motor motion or control of motor control. So next, I wanted to spend a few minutes talking about a question that's gained attention, I think, in recent years, or at least more so in this past decade, which is in resting state, we can have a lot of real neural state changes happening. You know, so especially if we're taking really long scans, like trying to study dynamics, people are going to drift between different vigilance states or even fall asleep. And this can have a big impact on our signal properties as well as connectivity maps. So what should we do to deal with that? I guess another illustration of this is from this article where they were looking at region to region connectivity states and their prevalence during alertness versus states of drowsiness. And I think that it's really cool that we can actually get different vigilance dependent patterns if we're trying to study brain states like vigilance. But if we're not, then this will be a confound in our study. And we can imagine there'd be systematic differences maybe between certain patient populations and controls. And so it's something that we do need to think about. So I guess, what can we do? I don't know if I have a great answer, except for that, like, there are certain measures you can record during your scan that can be an indication of someone's alertness. EEG is one, although it's very costly and difficult to do, you know, routinely, unless you're specifically doing EEG fMRI studies. And so one direction that people have been trying to take is, can we actually recognize the signatures of state change in our data from the fMRI data itself? And so this group looked at they found certain connectivity patterns that were characteristic of low and high vigilance, which may help us to understand when we're getting that kind of effect in our data. And we've been recently looking at spatial activity patterns that are linked with vigilance, and seeing if by characterizing the fMRI spatial temporal patterns, that can help us derive a measure of alertness from our data that would end up correlating with what we'd get from EEG if we had such recordings as well, which we did in the study to try to validate that. And so I guess the idea is that hopefully if we can understand the signatures of these state changes, then we can better model them in our analyses. So just in the last couple minutes, I wanted to mention something that's been, I guess, like a prominent issue and controversial over a number of years, which has to do with global signal regression. And so the global signal is the average time course of all voxels across the whole brain. And global signal regression is the procedure of calculating that average and then regressing it out of every voxel's time course. And this was shown to produce very clean looking maps if you do that, whereas if you don't do anything, then you can see this kind of blanket of positive correlations when you have a seed region. But this has been very controversial because several studies quickly showed that this procedure can distort true correlations and induce artificial negative correlations. And I put a few points now to try to summarize where I think some of the controversies arise. One is that even though we call this a global signal, it's not spatially uniform if we're deriving it from the average. So some regions may contribute more than others to the global signal. And then secondly, even though we often refer to global signal as if it's one thing, it contains contributions from multiple different processes like neural activity or physiology or vigilance. And from scan to scan, you may have a different proportion of things contributing. And so that means that if you regress out the global signal in one scan, it may actually be doing something a little bit different than if you're regressing it out in a different scan. And then there's also been some work showing that global signal may contain potential effects of interest, such as in biomarkers of certain disorders. And so, yeah, if we regress it out, then depending on what our goal is, we may also be removing some interesting differences. So a main alternative has been to try to use tissue or derive regressors from tissues other than the gray matter. So can we use white matter or CSF or large vessels, other areas in the brain that maybe are more likely to carry something related to artifacts and less likely to carry neural activity. Though this is also, there are interesting questions being raised in recent studies as well, because a number of groups are now showing that there may be neural activity in the white matter. I think that's still something debated, but there's some pretty compelling evidence from a number of groups now. So again, that is also maybe removing something of potential interest if we do that. And then multivariate methods and other algorithms have been proposed for reducing these kind of global or widespread confounds. Temporal ICA shows promise in separating multiple but distinct, spatially distinct widespread effects, or rather, like it's actually based on temporal ICA, so it's looking for temporal independence, but the spatial maps are allowed to look then more widespread, and so this may be able to identify components like RBT and vigilance better than spatial ICA, which tends to return more spatially sparse components. And then since a single global regressor may not be sufficient for removing certain widespread effects, Alex Vernita's group has also published this recent study looking at data-driven methods for extracting different widespread effects with different time courses, and showing how this may be more effective than global signal regression. So I think that now, like, we don't just have the question like, you know, should we regress the global signal or not? But I think the field is moving into really looking at what are the distinct widespread effects that are that are in our data. How can we parse those out and separate those from other things that we may want to study? Very last note is that there's efforts toward pipelines and other tools that are trying to help a resting state field be more reproducible. These can't remove all the tricky decisions for us, but they can definitely help our implementations become more standardized or more modular, and also provide nice ways of quality checking the data at various stages. So just wanted to also mention that. So finally, I guess resting state processing presents many choices. Many new methods for this are continually under development. Some processing steps and what we even regard as signal versus noise may be specific to our particular study. We can also study physiology from resting state data, for example, in which case we may not want to remove those kinds of low frequency effects. Understanding the sources of fMRI fluctuations can, I think, help to guide our processing decisions. And of course, when it's possible, I think it's nice to be able to record physio or other behavioral measures that can really help us when interpreting our data, because otherwise resting state, we really don't know what the subject is doing. And as a kind of quick plug for our recent work, if you have not been able to record physio but are interested in the respiratory variations, we're trying to use machine learning techniques for trying to extract respiratory signals and other physiological signals directly from fMRI data. So thank you so much.
Thank you. Thanks a lot, Cathy. It was tour de force. It was great. There was so much packed in those slides. I'm sure there would be lots of questions coming in for you, definitely. So we will take them towards the end, just like the other session. So probably we should move on to the next speaker, who is Sasuke Balman. So Sasuke did her MSc from ET at Zurich in 2013. And then she moved to Brisbane in 2014 to pursue her PhD at the Centre for Advanced Imaging at the University of Queensland. She then joined, after finishing her PhD, she then joined Martino's Centre for Biomedical Engineering at Massachusetts General Hospital in Boston, where she employed ultra-high field MRI to image the blood vessels of the human brain. And then she is now back to Australia, and she's now back to Centre for Advanced Imaging at the University of Queensland, where she is acting as a 7 Tesla National Imaging Facility Fellow. And she is currently implementing high-resolution functional and structural imaging for neuroscience research. So it's over to you, Saskia.
Well, thank you, Adil. So yeah, in this presentation, I actually won't talk so much about pre-processing because, as Max had said, you can't talk about it without also talking about the data. And I just see so many people setting themselves up for a world of pain. So I want to take the next 10 minutes to talk a bit about best practices for data acquisition before you even start your pre-processing. So when we look at noise and fMRI data in general, there are 2 big categories. One is the identically and independently distributed noise, which is the good noise because it usually matches the assumption of our models and it's pretty easy to deal with. And IID noise in MRI data is basically thermal noise from scanner electronics and the random motion of water molecules in the participant. The bad noise basically is the correlated noise, which unfortunately is the majority of noise we see. And this comes, as Cathy has described, from head motion, as well as physiology, but we also have noise introduced through image acceleration and system instabilities. And in the next couple of minutes, I will talk a little bit about how you can reduce the correlated noise in your data to make your analysis more easy. So actually, the easiest way to reduce correlated noise is to increase your IID noise. And that is simply achieved by higher resolution. And that's why some people advocate to go, instead of being in the physiological noise-dominated scheme or domain, to thermal noise-dominated domain, where you have more IID noise and you have to make less assumptions or deal with less correlations in your data. If that is not an option for you, because overall you will lose temporal signal-to-noise ratio, there are a couple of other things you can do. And starting with head motion, as Cathy has said, the best way is to minimize it as much as possible, but slow drifts are usually unavoidable. Motion spikes and head jerks can introduce really large artifacts, especially if you use accelerated acquisitions using SMS or multiband. And then you really have to think about advanced preprocessing by, for example, replacing a volume that is distorted in that way. And you also have to take into account the really long-lasting effects of this kind of motion in your data. Then really large displacement, especially rotations, can lead to changes in the B0 field. And that means the image distortion change. And you can no longer assume rigid body motion between the different volumes of your fMRI time series. And also your distortion correction will start to fail if the displacements are too big, because simply the distortion changes so much. So this is something to avoid as well. And the other thing is, basically, if you have a lot of noise during the reference scans, which are acquired in the first few seconds, so basically before the scanner actually sends a trigger, it acquires reference data, either ACS line if you use grappa, or sense maps if you use sense. And that can really introduce artifacts in very low TSNR in your time series. And because they are used, so the reference data is used to reconstruct the whole time series, you basically produce unusable data. And it's very important to instruct your participant that even if the task hasn't started yet, that they need to stay still, and also to warn them just before you start the scanner so they don't, or they aren't surprised and make some head movements because you startled them. And that brings us actually to image acceleration noise. So either grappa or sense, or simultaneous multi-slice or multiband. So if we look at in-plane acceleration, then you can see as the acceleration factor increases, if you just reconstruct your images in a traditional way using fast Fourier transform, you get this aliasing pattern. And sense or grappa help us then to unaliase this and get the full complete image back. But if you compare the top and the bottom row, you can see that here in the central part of the image, the noise is much larger. And this is indicated when looking at G-factor maps. So they basically tell you where most of the noise will end up, or where you have the highest noise amplification. And G-factor stands for geometry factor, and that characterizes the geometry of your coil. So depending on different coil configurations, these patterns will look different. Similarly, if we look in the slice acceleration, we have, again, an increase in acceleration factor here, and you can see how with increasing acceleration factors, artifacts start to creep in until basically the data becomes unusable. And that's kind of like one of the trickiest things if you set up a protocol to find, okay, what is a good mix, or what kind of acceleration do I need to answer my research question. And apart from just kind of like image quality themselves, people have also shown that because the noise through image acceleration is actually correlated, because it means pixels are not properly de-aliased, so you get signal from other parts of the brain folded into a location, and people have shown that you have false positive activation due to signal leakage between simultaneously excited slices. So what they did here, they basically ran a simple visual task, found a voxel with a really, really high t-score, and then kind of looked around at possible aliasing location and thought, okay, do we see a response there that is not visible when we use less acceleration? And this study was kind of like around the time when the human connectome project started, and basically led to actually the reconstruction and the update of the reconstruction algorithm for the simultaneous multi-slice technique. So that's why they then went back and had to reconstruct a large chunk of the data, because they noticed in the way it was done before that they introduced this slice leakage quite dominantly into the data. But nevertheless, here's an example from resting state networks, where you again can see really strong reliability at high acceleration factors. So you see here the multiband factor of 2, and they also have in-plane acceleration at 2. These really strange networks start to appear. So if your PhD student comes and says, oh, I found, this new part of the salience network that has never been seen before, but it only happens when I accelerate with a really high acceleration factor, then it's probably more an artifact than of neural origin. And this is also a recent analysis of the HCP data, because many people might work with it. And this looks at the HCP 100 release, where they looked at the motor task and basically asked, do we see slice leakage from this motor response here in other parts of the brain? And for that, they estimated, okay, where would we expect that? And then compared the right and left and right acquisitions. And they basically found that in 81 out of the 98 participants, they showed slice leakage. Does this mean that the protocol is per se a bad protocol? Well, it depends on your research question. So if we look at the standard deviation of the data, so this is a simulation, and here you can see the experimental results, and we see that overall in the experiment, okay, there's more noise overall, but they basically share the same pattern, and that is most of the noise amplification through the image acceleration is actually located in the center of the brain. As the human connectome project is mainly focused on the cortical surface, this is not a problem for them. But for example, if you're interested in basal ganglia or amygdala or some midbrain structures, then this is not a good protocol for you. And Cathy has already talked a little bit about breathing and cardiac activity. And I just want to touch on that again. This is a historical figure from 1993, where they already could record really, really high sampling rates simply by just acquiring one slice. With simultaneous multi-slice, now we can do that and still achieve whole brain coverage. And what you can see is that as we increase our sampling frequency, we really nicely can delineate breathing and cardiac activity. But you also see that we have these low frequency contributions up to about 0.2 Hertz. And if our noise were RID distributed, this should be a flat line. And in the title, you can see I already pre-widened that, but it was not successful. And that is because basically this really, really long tail here at higher frequencies, which the traditional models cannot address. So when you then use a more flexible pre-widening approach, then you can see that you can remove these low frequency contributions. But yeah, I want to point out that it's really, really important to still specifically regress out cardiac and respiratory activity, because pre-widening models cannot address this type of noise. And that brings us to the question, is it actually worthwhile to go faster? And here we see an example on a visual task with different noise models. And in the beginning, this was basically the kind of results the early studies show. Using an AR1 for pre-widening, we thought, okay, we saw double or 3 times the T statistics with lower resolution scans. However, if you use appropriate noise model that can correctly address the correlations in our data, then we see the difference is actually much, much smaller. And there's not that much to be gained if you have a very, very simple task design. Similarly, if we look at linear Pearson correlation for resting state, again, it depends really on what kind of preprocessing or noise model you use. You can see here that, for example, with pre-whitening, it's actually pretty detrimental to go to really short TRs. And you're better off maybe just going to, you know, multiband factor 2 or something. So in conclusion, we've seen we have some really cool techniques to acquire fMRI data with high temporal and spatial resolution. We, however, become increasingly susceptible to imaging artifacts, and we increasingly introduce correlated noise. The severity of the noise amplification depends on the coil geometry and the total acceleration factor. And in general, as a general rule, central or subcortical brain areas will suffer the most from TSNR reduction and signal leakage. And we have seen that serial correlations limit the immediate benefits. But for example, if you're interested in changes in HRF shape, then faster acquisition can be really worthwhile for you. This is the bonus slide because every now and then I see it in the literature and I just want to make sure that people go home with some correct knowledge. But it's true that SMS techniques are becoming increasingly important because they're crucial to decreasing acquisition times. They do not decrease geometric distortions from inhomogeneity effects. So SMS or multiband have nothing to do with geometric distortions from B0 inhomogeneities. And with that, I'd like to thank you for your attention and Lars Kasper, who inspired some of the slides and my group at the University of Queensland.
Thank you, Sasuke. That was great. I think you probably answered some of the multi-band imaging questions, but we can get back to them a bit more detailed. We have about 25 minutes to have this discussion now. So yeah, I mean, it was amazing. It was slides and data that you showed. So kick things off. I mean, if we start with something very simple-minded, if I were to pre-process my data, what do you recommend as diagnostics? Like if I'm not really into, you know, something which is very technical and you know, there's a lot of, you know, as we already see in just both of your slides about noise correction. So what do you recommend that once we have done pre-processing, what are the essential diagnostics that one should be looking at if the data is actually being pre-processed in a way that it's usable? Of course, some visual checks, but do you recommend any certain diagnostics that you usually do? So I mean, any of you, Cathy or Saskia?
So I'll give a few, so one is, it's nice to play your raw data as a movie. You can kind of just, I just like to do it, but you can also tend to see if any weird things are happening in your data. But as a more quick snapshot, this kind of carpet plot, I think is a nice representation. It's when you line up all these voxels on their all their time courses together in a matrix and look at it like a heat map. And then you can kind of see if there's any major ripples or anything that you're not expecting. ICA is actually a really nice kind of diagnostic too. Like if you run ICA, you can kind of look at various components and often like certain effects, like some of the multiband effects, I think will pop out in an ICA, and it just automatically gives you a lot of information about the structure of your data and how severe some artifacts may be. I'll let you add some. Those are a few that I look at.
Yeah, I would definitely recommend checking the spatial alignment. So I would say about 10%. So if you have like a group study, in 10% of the participants, probably the segmentation doesn't work out of the box. And then you have suddenly either just brains that are totally misshaped, and they then propagate onto the group level and can cause a lot of problems. So I would always kind of like, similar to watching your data, your raw data for each participant as a movie, also look at your raw data for the second level and make sure that, you know, every nose points in the right direction and all still have a brain shape.
Yeah, would you recommend looking at, for example, power spectrum of the signal, looking at if you know, just as you showed some of those, I think actually both of you showed this power spectrum of the signal, where you can see if the noise artifacts are still in the range, you know, if we are talking about resting state within the interest signal, like in the frequency range of interest. So it's looking at frequency domain, looking at those things would be useful.
Yeah, I've personally uncovered some weird stuff looking in the frequency domain, like a spike at a certain frequency that's it's not that large of amplitude, but it definitely is in your data. So if you're doing any kind of analysis of interest that involves a spectral analysis, like, you know, the power in some frequency band or the ratio of powers in other frequency bands, I mean, that's really important to catch those sort of things. So yeah, that's and if you're, you know, if you're trying to apply physiological noise reduction methods, you can definitely see how effective they might be by plotting your spectra before and after in your
Yeah, that's good. I mean, so one thing that I usually worry about, and probably that's where I get nightmares as well, which is that if I'm doing my pre-processing and I don't regress, because there's lots of signals that we're trying to regress out, we're trying to manage. Are we not regressing out the actual neural signal? So that's one of the biggest ways, that when I'm doing lots of aggressive you know, cleaning, as you say, of data, you know, I'm not actually regressing out what actually I'm interested in. Yeah, so that's my always sort of a mini nightmares that I get about. But probably there is a element of risk when you do it. But as you both said, it would be so having these diagnostics, looking at carpet plots, looking at frequency domain, power spectrum, spatial annealing, they help. So probably we should. There are quite a few more nightmares that I get, but probably I will just move on to the next question from the audience, which is do you recommend slice timing for multiband imaging? Any of you?
Depends on your TR, but I would say, I don't know, below 1 second, you're probably just in the range where you also have a lot of just variability in the people and in the shape of their responses to stimuli. So then kind of like the question is, do you start to just introduce lots of noise in your data because you're basically reinterpolating the whole data set, or are you kind of like better off with letting that in there?
Sure. What do you do, Cathy?
I would agree. So yeah, I mean, if your TR is very short, then it may not make too much difference. But I also haven't worked with multiband data very much myself yet, so I can't speak much from personal experience.
So I mean, there are few questions about multiband imaging. So probably yes. So so there's the next one, which is about rapid multiband imaging for fMRI. For Marcus Quay, have you looked into potential benefits for characterizing physiological noise with rapid board acquisitions? So Sasuke or Cathy, would you like to have your thoughts on that?
Well, I usually try to get rid of physiological noise. I didn't really try to characterize it, but yeah, if you kind of like want to look at the physiology, then it makes totally sense to go with faster acquisitions, because then you can like just resolve these kind of signals much more accurately. So yeah, but yeah, and kind of like if you just want to get rid of physiological noise, then yeah, it's, I think, even more important to do that if you sample fast, because the noise structure just changes so much.
Yeah. I mean, Cathy, you know, referred to it like if you know, they're going interest in looking brain body autonomic signals, then of course then these physiological noise that we are calling physiological noise become the signal of interest, and that could be something, but in most cases we just want to get rid of these. So there's a question from Maria Dibiase, and she's asking about to Cathy, can you please elaborate and talk about vigilance controlling the state in your paper in 2016 and the AML method that you, so yeah, that's exactly what I wanted to ask as well.
Okay, yeah, so we were wondering, can we see the vigilance effects in the fMRI time series themselves? And if so, then can we actually train a machine learning method to pull out from your fMRI data a time course that reflects continuous fluctuations in alertness over time? We actually initially did the study in monkeys that were in the resting state, and they'd actually get really sleepy like humans. And we were able to find a pattern, which also looks very similar in humans, of just a spatial map that seems to correlate very well with changes in alertness. So if you were to take, if you do have a concurrent measure like EEG, or like some measure of the eyelid open and closing like spontaneously, which we had in monkeys, then you can kind of correlate that against the fMRI data to get a spatial picture of what voxels are maybe increasing or decreasing their signals as a function of natural changes in vigilance. And so then what we then use that to project back to the scans to derive a temporal measure of vigilance, even if you don't have EEG. So yeah, so that was based on taking that pattern, and like if you have a new scan and you have like each volume of your new scan, we took the spatial projection or spatial correlation of that template map onto each volume of the new scan. So that produces like one value over time, which then traces out what we'd estimate to be a vigilance time course. And then we would validate that by comparing it to the EEG data that were acquired in the same. So EEG is another like kind of currently gold standard way of like tracking someone's alertness spontaneously. So
there's a question which is from which about the vigilance state, which is about can we use actually the eye tracking to capture the vigilance state?
Yeah, I think it's very powerful. And actually, like in the monkeys, we had more eye tracking data than, or rather eye monitoring data. So we didn't really track anything about gaze position, but we looked at how open or closed the eyelids were. And I guess you can also look at pupil dilation if you're interested in arousal changes. And so that can be, yeah, that can definitely be really powerful. And there's some papers on the correlation between spontaneous pupil dilation changes and fMRI signals, which are also show very, very widespread changes that are similar to what we see with the EEG. So yeah.
Okay, yeah, that's very useful. So I think I can now, it's time for me to throw in my another mini nightmare, which is about clinical populations. But for example, when you're working, for example, with aging populations, we know that aging can have a differential effect on vascular and neural signal. So how do we actually So how do we, so if we are working with clinical populations, how do we actually, when we are using functional MRI, we know how we can capture those effects because the bold signal itself is a mixture of both vascular and neural and noise. So how do you recommend we go about teasing out these different parts of the bold signal which may have clinical relevance?
Yeah, well, that's a very important question. And I mean, some initial thoughts are people have been using, for example, breath holding tasks and other kinds of vascular challenges where the idea is to kind of evoke a bold response that's related to vascular reactivity, but independent of the local neural activity. And so then in some way you can kind of calibrate regions like activity by by that measure. And so I think in the aging literature, it's been shown to be very important to do something like that. And then I've seen some papers from resting state data trying to get, using the amplitude of resting state fluctuations, a kind of proxy if you don't have breath holding data. I don't personally know how well it does compared to breath holding data, but they've shown some encouraging results. I'm trying to remember the authors. Yes, I will probably right after this. But I think that, yeah.
Sasuke, you have any thoughts on that?
Yeah, my study population are young, healthy PhD students, but I think it's really tricky for resting state because everything's kind of bundled together. But if you have kind of like a task, and then you can just, by using, you know, really straightforward simple block designs, you can just make yourself more robust against differences in hemodynamic responses and stuff like that. But just kind of like making your task simple and very robust to that.
Yeah, I mean, in a sense, so one possibility could be, you know, to use model-based work where you can actually have neurovascular coupling and hemodynamic model. And so that you can separate the bold signal into different compartments. So that's, but of course, but then it's not that straightforward. You have to have a generative model of how the data is generated, and then try to tease apart different factors. But I think this remains to be seen, because as we see that bold signal is a mixture of a lot of different components, and then making inferences based on that may, especially in clinical population, could be misleading. So that's to me another point that, you know, something that requires a bit more work in this area.
I think if I can just add one thing, what I kind of like often see is that, you know, elderly people just struggle a lot with a classical resting state. Just keep your eyes open. They just fall asleep after 2 minutes. And what people then do is, you know, show them movies or kind of like try to just keep them awake. And I think that's also, you know, quite nice and interesting point to take into account, how to actually keep your participants engaged in the task, which is much more tricky if you think about disease populations.
Yes, sure. Okay, that's then okay, so let's talk a bit more about task and resting. There's a question from Sahil Bajaj. He's saying that he's asking, is regressing out white matter and CSF for head motion correction a common practice in task-based fMRI as well, or is this concept from resting state? And similarly, regressing out signals from white matter and CSF for brain activation versus brain connectivity, is it recommended for both? So it is more about how do you process task fMRI where you want to regress out head motion.
Yeah, I have to admit, I tried it once to create additional regressors from white matter and CSF, and I regressed out my task. So I think it's really, really tricky, especially this was on 70 data, so we have so much more sensitivity, and you basically have to kind of like go and make sure that your masks are really, really well placed. So for me, it didn't work particularly well, and I prepared to acquire physiological noise directly and model it.
Yeah, this task correlatedness is very tricky. I've also seen a lot of, I think it's less commonly done for task data. I don't know, maybe just historically, but yeah, I think that you can see a lot of task correlated effects there, and also with head motion parameters too. And it's surprising, you have something like a visual checkerboard and you don't think that that's going to do some kind of major state change. And if you're monitoring breathing or head motion or heart rate, you can see sometimes very clear changes in the physiology when the thing comes on. Or we had a task where there's a tone every once in a while, and if we average this physio signal, like event locked around the tone, you can actually see like a very robust like heart rate and respiratory change. And that was showing up in our task map too. And so then I think that like if some of those effects are temporally decoupled from the task, then you can include them. Like sometimes that some of the respiratory effects are delayed, like, and that may be more true for like physio regressors than for like brain derived signals like white matter. And so then you may be able to clean it up a little, but yeah, this is one of those things where like, if they're correlated, it's really hard to really say there's a right answer, except for it's good to know if they're correlated because that's gonna impact how you interpret your data. So it's better to know than to, I guess, report something and then find out that they were just had task correlated like everything, so.
Yeah, I mean, it's complicated, but I think the issue is this, that with a resting state, these micro head motions and all that, and because they are correlated with the default mode network in one of your slides, Cathy showed, but with resting, with tasks, first of all, you can average across trials, and then you are taking differences between those as well. So if there is a background, head motion or physiological noise, if you're making contrasts and averaging those, so probably you cancel out some of those. But I think, as you said, there is less of that very task. But I think some of what I have seen recently more common is this, that people have started to use white matter and CSF more in task. Because there's more awareness probably as things have moved on from resting state, that it may be something that needs to be at least think about a bit more carefully. So let's change gears and now there's something more towards functional connectivity analysis. There's a question about what do negative correlations in pairwise FC represent, and do you think they should be treated separately to positive correlations? So that's probably not more of a pre-processing, although it may be related to the GSR. Some of the Cathy was talking about, which is more about the interpretation. But what are your thoughts on that?
Yeah, I mean, I would say that. So I maybe in general, not really sure what negative correlations may reflect. I mean, one of the big findings is that the default mode network and task positive network tend to be negatively correlated, and then the neuroscience, to my knowledge, the neuroscience explanation is something, or at least theory is that there may be some kind of like interaction between them, and that they're kind of like, like, what is the word? Like they're like regulating each other in some way, or when one is up, the other one is suppressed because one is an external versus internal balance. So I think that I can't really speak much to the neuroscience interpretation, but I always feel that like for our analysis, we should treat them separately because they are about opposite sign, and that I think that, I mean, it may depend on what your analysis is actually. So I'm kind of rambling, but I think that I usually like to treat things separately if I'm not sure. I think, yeah. And it's also, I guess, important to see, like, are they really negative correlations, or is there a phase lag between them that could kind of like manifest as a slight negative correlation? So if you can plot the signals and check them, then that's good.
Yes. So yeah, I think it is more about, as you said, about interpretation of these and what they actually biologically, what do they mean, which I think still needs to be fully understood. So again, we change a bit of gears, and there is a question from Kavisha Fernanda, which is who is talking about how do you, I think probably, so it's automated tools to determine whether registration was faulty. So any thoughts about registrations?
I think fMRI prep gives you kind of like a visual result where you can see if the alignment works, at least on a single subject level. But I'm not aware of anything kind of like for group studies.
Sure. Any thoughts from you, Cathy?
Yeah, I mean, I'm not really aware of many things that is beyond that. You know, if you can see the outlines superimposed together, that's really helpful. And sometimes it's like part of the brain is not, if you're doing nonlinear warping, sometimes you can see weird stuff, like part of the brain is not aligned, but like there's a bulge or something, but like, I don't know. Yeah.
Sure. I mean, we don't have a lot of time remaining, so probably we take another one or two questions, and then I think there's another pressing issue about global signal regression that we have touched upon, Cathy. So there is a question about that there's a recent comparison of different pre-processing pipelines that show GSR seems to improve network detection. For example, Parks 2018 and Strict 2017, even in addition to ICA methods. So basically the question is, how do you bring these, so there is an opposing views on this, and there's no convergence on what to do and what not to do. And I think you did some advice, few recommendations. So would you like to take up a bit more on this and tell us what you think about this?
Sure. Yeah. So I'll have to go see those papers because I may have read them, but I don't remember the details. But I think it kind of depends what they were comparing global signal to versus. So are they doing global signal versus nothing, or like global signal versus like some other kinds of regressors? And I mean, I think I can definitely imagine cases where it will do better than nothing. And I guess here you said in addition to ICA methods. Yeah. I mean, depending on what those are. And I think, yeah, it depends on, well, what were the characteristics of the data sets that they applied global signal regression to? So were there people with were the subjects, did they have a lot of low frequency changes in physiology or a lot of vigilance changes? And maybe it kind of catches a lot of those and you may see an improved effect. I think my personal preference is to try to model those effects more specifically rather than to use global signal regression, just because, I mean, there's some uncertainties in terms of what it's doing exactly. But yeah, I mean, I've seen a lot of cases where it does seem to clean up the data better than doing nothing. And so, yeah, I can see that maybe it's not contradictory, but yeah.
Yeah, I mean, we are running out of time, but there's another very interesting question that I would like to have some thoughts from you, from both of you, Sasuke and Cathy, which is about the test, which is reliability. So I think that's a pretty big, probably requires a whole section on the test, which is reliability, reproducibility of vesting state functional connectivity literature. So the question is about how do you act? Are there any recommendations that you can take certain steps during your pre-processing that would improve the test reliability of some of these measures? So any of you, Saskia or Cathy, would like to talk a bit about?
Probably the other way around, kind of like making sure that the data that goes in is as similar as possible. So yeah, try to kind of like catch the people in the same state of mind and make sure they're awake in both scans and make sure they move approximately the same in both scans. We've seen how caffeine changes all kinds of stuff. So I think just controlling really, really well when a person goes into the scanner on all of these variables.
Yeah, and because state differences, they can be pretty major. It's kind of surprising and also kind of scary because you're wondering like what other things that we don't know about that we should be controlling for. Which is also something that like gives me nightmares sometimes. Like just don't know everything about it. But yeah, I mean, and there may be real. If we are scanning people at different times and different conditions, then we would expect the brain would not be exactly the same. So yeah, I mean, I guess the more we can control for, then.
Yeah, I think the main premise is that function is dynamic, and probably we set a pretty high bar when you compare, like with the structural measures, you get a really high tested reliability because it's not dynamic. It's at least for time scales that we're interested in, it is static. While function is another beast, as you both said, there's there are lots of different things going on. Even the time of the scan that you have done is morning or afternoon or evening, what you had a coffee before or not. So these things you can have an impact on all those things. So probably there's no one silver bullet here, but probably and as Sasuke said, the idea would be that you make sure that whatever you are doing, you're doing in a way that is consistent across the data sets. Okay, so probably we are running out of time. So we would wrap it up here. It was wonderful. I mean, session. There is so much that one can talk about, and it leaves you with like having a bit more time. But thanks a lot. You both had given us a lot of insights, and I'm sure that would be useful for many of us. So with this, again, thank you, and I would hand it over back to Alex to wrap up the session. Thank you both again. Thanks a lot.
Thank you.
Thanks very much, Adil, and thanks to Katie and Sasuke. In fact, thanks to all our speakers, Katie, Sasuke, Maxime, and Maria, and also our moderators, Adil and Andrew. I think it's been a fantastic session. I've learned a lot, some really excellent work, and even more food for thought, even though I've been working in this area for quite some time. Thank you all for joining in. Thank you for all the questions. I really do think it's been a great session. And moving into the new year, we will send out notifications about new webinars that we will be planning, and we will, we intend to continue doing these throughout the year. And as I mentioned earlier, as things become clearer about the prospects of an in-person meeting later next year, we will make we'll send out notifications about that. But in the meantime, thank you all for tuning in. Have a wonderful Christmas and New Year break, and we'll see you in the new year. Thank you.